Hello everyone. Today we are going to discuss about some important questions regarding AP Set Life Sciences. So this topic covers some questions in the topic developmental biology, and in developmental biology chapter you can see basic concepts of development. That is the first topic. In that topic some of the questions which may be asked in the AP Set Life Sciences or any set examination which is conducted from the state. So see the first question. Identify the correct sequence of progress process in animal development. So the option A is cell differentiation, regional specification, morphogenesis, growth. Option B, regional specification, cell differentiation, morphogenesis, growth. Option C, morphogenesis, regional specification, cell differentiation, growth. Option D, regional specification, cell differentiation, growth and morphogenesis. So let's see what is the answer. Answer is B, that is regional specification, cell differentiation, morphogenesis, growth. So this is the correct sequence of process in animal development. How can we say this means? A little bit explanation I will give you. So during regional specification, cells are assigned a specific fates based on their positions within the embryo. So whenever the cell differentiation will takes place, so this is nothing but regional specification where they will take the positions in the embryo. After that, cell differentiation will take place. This cell differentiation is the process by which unspecialized or pluripotent cells will become specialized and take on specific functions. So during this cell differentiation, cell will take different functions. And coming to the next in the sequence, that is morphogenesis. So morphogenesis is responsible for forming the body plan and arranging the organs and tissues in their proper positions. Then growth. So growth is the last in the sequence. So what happens in the growth? Growth involves an increase in the number of cells through cell division and an increase in the cell size. So like that, the sequence of process in animal development will takes place. Next. Next question is morphogenic field reflects. So morphogenic field reflects option A developmental potency, option B polarity but no axis, option C break of communication, option D developmental fate. Answer is D. So morphogenic field reflects developmental fate. So if you see the explanation, morphogenic fields are hypothetical organizing fields that play a role in embryonic development. They are thought to guide the differentiation and development of cells and tissues in the embryo, ultimately determining their developmental fate. So this is nothing but morphogenic field reflection. Next, embryonic stem cells are useful in regeneration therapy. These cells are also useful in transgenics. What are they? So option A pluripotent, option B totipotent, option C unipotent, option D omnipotent. Our answer is option A, that is pluripotent. So what is pluripotency means? Embryonic stem cells are a type of stem cells derived from the inner cell mass of the early stage embryo known as the blastocyst. So we can find the embryonic stem cells in the blastocyst. So these cells have the unique property of being totipotent. So this totipotency you can observe normally, but whatever the cells which are embryonic stem cells, generally they are pluripotent, so they can differentiate into any type of cells, so which can be give rise to the new organ, and that organ may grow. Next, fourth one, that is differentiation process in life cycle of animal is a specialized state, option B unspecialized state, option C common state, option D uncommon state. Answer is A. So the differentiation process in the life cycle of animals is the process by which the cells become specialized to form specific functions. During development and growth, cells undergo differentiation, acquiring unique characteristics and that allow them to perform specific characters. So contribute to the function of various tissues and organs in the body. So that's why option A, specialized state, is the answer. Next question. Renewability, pluripotency, and ability to differentiate are the properties of. So, option A, endocrine cells, option B, endodermal cells, option C, oocytes, option D, stem cells. So, our answer is option D, that is stem cells. So, stem cells possess the properties of renewability, pluripotency, and the ability to differentiate, making them unique and crucial for various biological processes and potential therapeutic applications. That is the capacity of stem cells. Next question. Fate mapping with vital dye for amphibian eggs was done by Oct 1929, Mangold 1931, Spiman 1930, Von Bohr 1927. Answer option A, that is Oct 1929. So Walter Ocht was a Swiss embryologist who first used vital dyes to trace the fate of different areas of amphibian eggs in 1929. He used agar chips soaked in vital dyes to mark specific regions of the embryo and then observed the subsequent fate of the stained cells. So this is about fate mapping with vital dye. The next question. Which of the following is not an internal factor of differentiation? Option A, polarity. Option B, inductive effect. Option C, mutual incompatibility. Option D, cytoplasm. Answer D, that is option cytoplasm. So there are actually cytoplasm. On the other hand, it is not typically considered an external factor of differentiation because it is a fundamental component which is found in the cells and it is not a factor that directly causes differentiation. Differentiation usually involves changes in gene expression, cell signaling and other molecular processes. While cytoplasm is a cellular structure that contains various organelles and molecules. So that's why it is not an internal factor of differentiation. Next, in birds and reptiles, primordial germ cells originate from option A, epiblast, option B, thymic epithelium, option C, germinal crescent, option D, embryonic allantois. Answer is option C, germinal crescent. So, generally, so generally, birds and reptiles, primordial germ cells originate from germinal crescent. So, in avians, like in birds and reptiles, development of primordial germ cells or special cells give rise to the future reproductive cells, that is sperms and eggs. So, this germ cells may be specified at a location called germinal crescent. So, it is a specific region within the embryo of birds and reptiles and it is typically found in the early stages of avian and reptile development. Next question. Question number 9. Stem cell factor is produced mainly by option A, stem cells, option B, thymic epithelium, option C, bone marrow stromal cells, option D, megakaryocytes. Our answer is option C, bone marrow stromal cells. So, stem cells factor are known as c-Kit ligand or steal factor and it is a protein that plays a critical role in the regulation and maintenance of hematopoietic stem cells. So, it is produced mainly by bone marrow stromal cells. Question number 10. The initial cell division during development is anticlinal, periclinal, random, oblique. So, what is the answer? Option A, that is anticlinal. During the early stages of development in plant embryos, the first cell division that occurs is known as anticlinal cell division. This plays a crucial role in establishing the basic body plan of the plant embryo. It sets the foundation for subsequent divisions and tissue differentiation. Then, identify the correct statement with regard to embryonic stem cells. Option A, they are not derived from inner mass of blastocyst. Option B, cannot proliferate into different cell types. Option C, they can generate primitive ectoderm. Option D, they are not pluripotent stem cells. Option is C, they can generate primitive ectoderm. So, embryonic stem cells are having the capacity to generate primitive ectoderm because they are derived from the inner cell mass of the blastocyst and have the capacity to differentiate into cells representing the primitive ectoderm. So other germ layers, primitive ectoderm is the layer of the cell that forms during early development and gives rise to many different cell types including skin, hair and nerve cells. Then, the establishment of anterior, posterior, dorsal, ventral body axis is called as. So, what is the formation of dorsal, postal and ventral axis formation? So, what is the option here? Option A is pattern formation, option B morphogenesis, option C differentiation, option D growth. So option A is the correct answer, that is pattern formation. So it is nothing but generally pattern formation refers to the process by which the body axis such as anterior posterior, that means in common we can call as head tail, and dorsal ventral, that is back belly axis are established during the early stages of embryonic development. This process is crucial for the organization and arrangement of cells and tissues to give rise to the proper body plan of an organism. Next question. Basic principles of embryonic development were pronounced by von Baer, Weismann, Haeckel, Morgan. So, Carl Ernst Ohnbar. Option A is the correct answer, that is von Baer. So Carl Ernst von Baer is the person who first uh, described these basic principles of embryonic development, and he is a 19th century biologist. Significant contributions he made in embryology. So he formulated set of fundamental principles in embryonic development also, and those principles we call like von Baer's laws. Okay. And then next question. The process by which cells become structurally and functionally distinct during development is called as option A differentiation, option B determination, option C specification, option D both A and C. Option A is the correct answer, that is differentiation. So differentiation is a fundamental process in the development of multicellular organisms including plants, animals, and it involves changes in the gene expression and cell morphology that leads to the formulation of different cell types with specific functions. Next question. In Drosophila, the phase of early blastomeres is determined through option A autonomous specification, option B conductional specification, option C syncytial specification, option D maternal specification. Option D maternal specification is the correct answer. So when happens this means in Drosophila during oogenesis, the mother places certain proteins and RNAs known as maternal determinants into the egg's cytoplasm. As the zygote undergoes its initial cell division, these maternal determinants become asymmetrically distributed among the blastomeres, leading to the specification of different cell fates based on their positions and the localized presence of these molecules. Next question. In chick, development of wing feathers, tail feathers and claws depends on epithelial specificity conferred by induction from mesenchymal components from different sources of the dermis. This may be attributed to option A, autocrine interaction, option B, regional specificity induction, option C, genetic specificity of induction, option D, autonomous development. Option B is the correct answer, regional specificity of induction. So what is this regional specificity of induction? This is the key aspect of the scenario. It is different mesenchymal components from various regions of the dermis provide specific signals to the epithelial cells. These signals result in the differentiation and development of different structures depending on the location of the interactions. Next question. Embryonic stem cells are obtained from inner cell mass of as a precursor. Option A, epiblast, option B, hypoblast, option C, tonoplast, option D, blastocyst. Option D is the correct answer, that is blastocyst. So the blastocyst is an early stage embryo that forms around 4 to 5 days after fertilization in mammals, including humans. During embryonic development, the fertilized egg undergoes multiple cell divisions to form a hollow ball of cells called as blastocyst. We'll move on to next question. Shinya Yamanaka received the Nobel Prize for the discovery of option A, induced pluripotent stem cells, option B, oncogene, option C, Okazaki fragments, option D is cancer stem cells. Option A is the correct answer, that is induced pluripotent stem cells. We'll see who is this Shinya Yamanaka. So Shinya Yamanaka, a Japanese stem cell researcher, was awarded Nobel Prize in Physiology of Medicine in 2012 for his groundbreaking discovery of induced pluripotent stem cells. So these are a type of stem cells that are generated by reprogramming adult somatic cells such as skin cells or blood cells to exhibit the pluripotency. And next, Drosophila displays the method of embryonic development. Option A, ametabolous. Option B, hemimetabolous. Option C, holometabolous. Option D, porometabolous. Our answer is option C, that is holometabolous. Holometabolous is the method of embryonic development in Drosophila. So this development is also known as complete metamorphosis, and it is a type of development seen in certain insect species including Drosophila, that is fruit flies, and it is characterized by series of distinct life stages, that is egg, larva, pupa and adult. Next. So this is a match the following. You can see here list 1 is having unipotent, oligopotent, pluripotent, multipotent. And here in the list 2 will be having some options you need to match. So what is unipotent? It is nothing but cells that differentiate into any one cell type but capable of self-renewability. That is called as unipotent. Then coming to oligopotent, cell is more restricted than multipotent but can still differentiate into a few closely related types. Then pluripotent, cells that can differentiate into cell types of an adult organism. Multipotent, cells that differentiate into multiple different but closely related all types. So this is unipotent, oligopotent, pluripotent, multipotent. Very, very important. Okay. So with this we have completed our today bits regarding developmental biology basic concepts, and in the next video we are going to discuss some more important bits regarding developmental biology. For more videos, please subscribe our channel KNK Knowledge Hub. Thank you.